Welcome to solverfy.
All right, we are given a instance where a pre-mRNA, so up here, is processed into mRNA, and we are given the normal process. So on the left-hand side is what the normal resulting mRNA would look like, and on the right-hand side is the mutated version of that pre-mRNA. And in the step going from pre-mRNA to mRNA, this is where splicing occurs. And splicing is the removal of introns and or unnecessary exons. So if you notice here on the left-hand side, number 2, section number 2 has not made it down, and section number 4 has not made it down. Whereas on the right hand side, it's also missing section number 3. Okay, so missing section 3. So that is the mutation. Now, you need to know a little bit about how this happens in order to be able to answer this question. So, Tata boxes they are promoter regions, and this is where promoters bind. So it is basically a region of the DNA where it is a repeat of TA, 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 TA. And that is a motif that the cell uses to recognize promoters. So that's how promoters know where to bind. There are also CPG islands, which is repeats of CG, CG, and the P in between refers to the phosphate that connects the nucleotides. And this is where DNA is methylated. So that is the motif that the cell recognizes for methylating DNA. Now, what is the motif for cutting out exons? How does the cell, because it's easy for us to just draw lines and be like, oh, this is one exon, that's another exon, but how does the cell know? How does the cell demarcate these different exons? And those are GU and AG sites. And basically, GU is denoted to is used to denote the beginning of a section, and the AG sites are used to denote the end of a section. So basically, you would have switch color pen. You would have a section of the DNA where it'd be like, I'm sorry, there should be a G, where you would have GU, perhaps on a repeat, and then gene, and then AG. And then the cell recognizes, okay, from here to here is one intron or one exon, and either keep that or excise that. Okay, so the way that the cell knows where introns or exons begin and end in order to splice them out of the pre-mRNA is through these GU and AG sites. Okay, so let's look at our answer choices. First one is mutation in the GU-AG sites of introns produced a non-functional protein. That sounds correct. So I think A is a viable answer. Choice B, a transversion mutation in the introns led to alternative splicing, producing a functional protein. Transversional mutation, so let me write this down. Transversion is when you switch from purine to pyrimidine, or vice versa. So basically, when you change the type of nucleotide base that there is in the DNA sequence, it's called a transversion mutation. Now, this answer is also not wrong, but there's a level of specificity included in there that is not warranted. this GUAG site doesn't have to be so a mutation in this GUAG site doesn't have to be a transversion mutation in order for it to make it dysfunctional. So A is a more complete answer. Choice C, a transversion in the mutation. So again, it's talking about transversion mutation, so I would eliminate C for the same reason as B. Choice C is D. Choice D is transition mutations in the introns. Transition mutations. So transversion is when you go from purine to pyrimidine. Transition is when you go from purine to purine or from pyrimidine to pyrimidine. Okay, so you're not changing the base pair. Again, that's a level of specificity that's not warranted. So the most complete correct answer here is choice A. Just a mutation that disrupts the GUAG site will produce a non-functional protein.